In general, a tumor grows when we have one of two types of mutations. Either a mutation that's telling the cells to grow out of control, or the opposite, where we've got a mutation that is tamping down the genes that tell it not to grow. And so with a brain tumor, with any tumor, when we have these mutations, it's disrupting the normal flow of growth and apoptosis, or natural death.
Normal cells, cancer cells, are fascinating that every 8 hours when they divide, there's billions of DNA that needs to be repaired. When they're replicated, there'll be mistakes that are made, and the DNA damage response pathways are responsible for fixing those errors. Now, normal cells don't divide that often, so they don't make a lot of errors, but cancer cells are rapidly dividing and rapidly growing, and such that there are many mistakes that occur, and the DNA damage response pathways help them survive through those replication cycles. Often they have defects, or they have dysfunctional ways that they run those pathways. And central to that is one protein called PARP that's implicated in brain tumor survival.
If we can target those proteins that aren't supposed to be there, then we've got a targeted therapy, and we can fight what we call the driver of the tumor and make it stop growing.
In about 2015, when our lab had just started at Yale, about 3 years into my faculty, we made a series of discoveries where we found that certain tumor-associated mutations found in adult, but more often pediatric and what we call the adolescent young adult population, or the AYA population, we found that there was mutations specifically in those tumors that caused DNA repair defects. What was very interesting is that in my early faculty, I won a CureSearch Young Investigator Award. And this was a 3-year award that really provided the infrastructure for us to make the discovery, which was the link between IDH mutations and DNA repair. We were able to treat cells that have the IDH mutation with a class of drugs that are FDA approved called PARP inhibitors. PARP inhibitors work against tumor cells that have this defect, and we realized that we could use the FDA-approved PARP inhibitors and try them in young adult and pediatric patients that have these IDH mutant gliomas.
So you've got a very small population with very specific mutations that need to be studied on their own.
As you can imagine, very difficult to get funding for this sort of trial. And that's when we went back to CureSearch and were able to take advantage of the CureSearch Catapult Award. And this award allowed us to essentially take this bench discovery and move it directly into the bedside.
The trial that Dr. Bindra and I worked on has many components to it. And so it takes many different experts, and it takes a large amount of funding sources to make it all come together.
We're always grateful to our donors, the foundations that support this really difficult work. And so we looked at other consortiums like the Pacific Neuro-Oncology Consortium, who actually then allowed us to run our trial across a broader network of sites, and they also provided their own funding. And so working together with the CureSearch and the PNOC group, we were able to do a national trial and enroll a fairly large number of patients in a short period of time.
I think this trial is helping us be more cognizant of specific mutations and unique targets that we can look at in adolescent and young adult tumors. Priscilla was a really interesting and unique case. Her tumor was a high-grade tumor. At the same time, it carried a specific mutation that we felt that we could target.
Obviously, I was dealing with this new diagnosis that came out of nowhere. It was an inoperable tumor. So from the beginning, they told me we're going to start radiation. I was 23 and living on my own. And where I needed to get radiation was 2.5 hours away. And then during radiation, they said, a few days after you finish radiation, we're going to start the chemo. But then I wasn't even finished with the first cycle when I had to go into the hospital, and after testing, they saw the bad brain inflammation. And so thankfully we reevaluated, and I was referred to you to over here.
So the initial work out of Bindra's lab informed the trial. The trial has given us confidence in treating patients with this particular class of drug in this particular mutation. And so when we saw Priscilla's mutation, we had the confidence to go ahead and try this treatment course.
Feeling more confident in the care I was receiving made a big difference, because even though I was sick, even though I was not feeling well, as long as I felt like we were truly fighting the disease, then I saw an end in sight, and that helped. That helped a lot.
We've gotten control of Priscilla's tumor. She is still tumor-free, despite the fact that she's off medication, and she's doing great.
So even though we had treated 50, 60 patients in this trial, we already saw early evidence of efficacy and response, which then allows us to treat patients with the same tumor. And that's what's the beauty of these small trials having a massive impact on pediatric cancer research.